Ever wondered how cells eat and drink? Let's dive into the fascinating world of endocytosis. Endocytosis is the process by which cells take in materials from the outside environment by engulfing them in the cell membrane. There are several types of endocytosis, each with its unique mechanism and function. First up, phagocytosis, also known as cell eating. This is used by cells to take in large particles like debris, pathogens, or dead cells. Immune cells, such as macrophages and neutrophils, are experts at this. The cell membrane surrounds the particle, forming extensions called pseudopodia. These pseudopodia close around the particle, creating a large vesicle known as a phagosome. The phagosome then fuses with a lysosome, where digestive enzymes break down the particle. Next, we have penocytosis, or cell drinking. This process is used for the uptake of fluids and dissolved small molecules like nutrients. It's non-selective, meaning the cell takes in whatever solutes are in the fluid. The cell membrane invaginates, forming a small vesicle that traps the extracellular fluid. These vesicles then move into the cell for further processing. Receptor-mediated endocytosis allows cells to selectively take in specific molecules such as hormones, nutrients, or ions. These molecules bind to receptors on the cell membrane, forming a receptor-ligand complex. The cell membrane invaginates, forming a clathrin-coated vesicle that transports the molecules inside the cell. Caveolae-mediated endocytosis involves small, flask-shaped invaginations called caveolae. This pathway often transports smaller molecules like lipids, proteins, and certain toxins or pathogens. The vesicles, coated with proteins like caveolin, trap specific molecules and transport them to different parts of the cell. Lastly, macropenocytosis is a non-specific process used mainly for large fluid uptake. This is often observed in immune cells like dendritic cells. The cell membrane extends large folds known as membrane ruffles that engulf large volumes of extracellular fluid. The fluid-filled vesicle, or macropenosome, enters the cell for processing. In summary, each type of endocytosis serves a specific purpose. Phagocytosis engulfs large particles. Penocytosis takes in extracellular fluid. Receptor-mediated endocytosis selectively uptakes specific molecules. Caveolae-mediated endocytosis specializes in small molecule uptake, and macropenocytosis captures large amounts of fluid. These pathways allow cells to interact with their environment, acquire nutrients, clear waste, and maintain homeostasis. Fascinating, right? That's endocytosis, the cellular way of eating and drinking.